Thank you for inviting me, particularly Lisa. Well-organized conference. I've really enjoyed everything so far. Thank you. Well, today repetition's the mother of learning, so a lot of what Chris said is going to be repeated by my talk, and he spoke about the places that will be involved. This is the clinical center. This is the hospital at the NIH, and then this is where my office is. And then we have pediatric neurosurgeons. Dr. Goldstein is a neurochemist, and I'm involved there, and other people too. So we're very happy to be included in the group of sites for the trial. So the trial uses a vector that has tropism, or it's attracted to the brain, so which is perfect. And it limits gene expression in other body sites because we don't want to have toxicity in other body sites. And Dr. Edward Oldfield, my old boss, he's moved on now, but he developed convection-enhanced delivery at the NIH. So it has a good safety record, and we're very confident in its use there. Now, there are several trials with AAV gene therapy that have been going on, including Dr. Hu's study here. That's included in all the trials. And then there are other ones with GAD expression. That's just another enzyme. And this trial's been completed, but at the NIH, we're using a trial, not with nurturin, but with GDNF. And I'll tell you, that has some parallels with the trial with AADC, AAV2 AADC. So I'm going to just describe some of that trial to you because we'll do the same kind of stuff at the NIH. So as Chris said, with the AADC mutation, you have the mutated gene, and then you have the mutated enzyme that can't really bring dopa to dopamine. So what we do is we insert a new gene with the vector, and then we bypass the block, and we make the dopamine. So that's the idea. If gene therapy works, that's how it's going to work. And Dr. Hu, as Dr. Bankiewicz said, he showed on the PET scans that he can increase dopa uptake, dopa production when he puts in the gene therapy. So that's evidence that he was able to substitute some AADC gene and that it actually worked. So these viruses are very popular as far as gene therapy. Some of the other viruses were very immunogenic. In other words, they'd be rejected. The brain is privileged. So if you put one of these gene vectors in the bloodstream, you're going to have an immune reaction. If you put them in the brain, they're sort of protected from being rejected. And that's a good thing if you want to have gene expression in the brain. So it doesn't trigger much of an immune response. And this is one thing that we're doing with the GDNF trial. Like if you have deep brain stimulation, you can turn it on, you can turn it off. Once you use gene therapy, the gene's always on. So pretty much if you have a complication, that could persist. If you have a good effect, that should persist too. But that's just, we can't take it back, like a lot of things in life. All right, so here's the size of a virus. Like here's a young child, housefly, red blood cell. You can see these on a microscope, but below that, they're so small you can't see them on the microscope. Here's the size of a hepatitis virus, and here an AAV2 is even smaller, so it's very, very small. But it has just enough room to put one gene in. So you put in the AADC gene in the virus, and then it's a virus, so it can't reproduce on its own. It has to reproduce in a cell, so you put it in these producer cells, and that makes the viral vector. And as Dr. Bankiewicz said, it only has one gene, and in this case, it's the AADC gene. And they're all packaged. These are called, they're packaged genes. So, as Dr. Bankiewicz said, here we're going to put it in the substantia nigra and its neighboring structure, ventral tegmental area, and then that projects into the caudate and putamen, and together those are called the striatum in the brain. So even though you put it in a relatively localized area, you have projections broadly to a much larger area. I went through that before. Okay, so children that are coming to the NIH, they would stay at the Children's Inn. So as a surgeon, it's like, hi, glad to meet you, and then we do the procedure, and we'll follow somebody for a week, and then we take the stitches out, and it's like, it's been nice knowing you, and they move on with life, and if they've had a good result, then they're happy, we're happy. But with this trial, it's much different because it's going to take a while for the gene therapy to activate itself, generally 3 to 6 months, I think, in Dr. Bankowitz's animal studies. And so the patients, we're going to watch them, we're going to examine them carefully, and they'll have to be followed by a pediatric neurologist at the NIH. But they'll stay at the Children's Inn. This is very similar to what a Ronald McDonald House in the U.S. I assume there's the equivalent since McDonald's is multinational. I assume that you have those here too. And it's just a hotel, not as nice as this one, no golf course, but it's a nice hotel. And there's areas, common areas where you can cook and so forth, and there are playgrounds outside. It's a little colder there in Washington now, but in the spring, it'll be like that. All right, so the objectives are, as Dr. Bankwitz said, safety and tolerability of the treatment. And the secondary would be if we can restore the gene and if motor function improves and if quality of life improves. That's what we're looking for. All right, so this is why I flew over here, because some people wanted to know about the surgery. So patient comes to the hospital, has all the tests. If they're good candidates, then they go into our operating room that has the MRI scanner. And we have anesthesiologists like other hospitals, and they put the child to sleep. Then the child will go into the scanner. This is the front end of the scanner, but where we operate's behind here. And so we put these sterile drapes here, and the head will project out here, and that's where we'll do the surgery. So I want to just draw parallels from a gene therapy trial we're conducting now. Dr. Bankwitz is doing AADC gene therapy now at UCSF in Parkinsonian patients with Parkinson's disease, but those are generally older adults. This is another trial that we're doing at the NIH. It's with GDNF, which is a neurotrophic factor. It keeps nerve cells from dying prematurely, and it makes nerve cells sprout. In other words, they can make more connections. All right. So what we use this aiming device. It's plastic. It's MRI compatible. It's not going to get stuck onto the magnet like iron would. And what we do is we line up this catheter guide, and we line it up with our target in the brain. We put a grid that has, it's just a piece of plastic and has a little bit of gadolinium in all these little cells. So we put that on the head, and then we determine what's the safest place to enter the skull and the brain. We don't want to hit any blood vessels going to the target. Then we choose a target. It'll be the ventral tegmental area or the substantia nigra, and that will be, and then it's just geometry. You have 2 points, and 2 points are connected by a line, and the computer can figure that out, and it determines the trajectories. So this is all virtual now. This is all a picture, but we haven't done anything yet. But see, here's one of those places on the grid, and we'll say, well, okay, we'll take this point, and that will help us get right to our target down there. So now we're going into the cutting part, okay? So here we take off the piece of plastic, and we mark the skin where we're going to open the skin. here's the top of the head. So we open the skin, and then we make a hole in the skull, and then we put this little platform down and connect it to the skull with 3 screws, and then we attach the aiming device to it. And as I said before, this is where we put the catheter. So we have to line this up. It's just like you have a rifle, and you're sighting. So you want this to aim right at the ventral tegmental area and the substantia nigra. And we have these little adjustment screws here, and we can just make very, very fine adjustments to line up the catheter guide with the target. As shown here, here's where we're aiming and we're lining everything up. So, and then after we line it up, then we pass the cannula or catheter, and then we start infusing the gene therapy. Now, viruses are small. I already told you can't see them. So what we put in the solution, we put in gadolinium, the same type of contrast that's normally used on MRI when you have a scan and you're looking for a brain tumor or something like that, but it's very dilute. And so that way, and there have been a lot of laboratory experiments, that way we can see how far the solution is going, and we know from experiments that the solution goes as far as the virus goes. We selected a type of gadolinium that distributes the same distance as the virus, or the viral gene therapy. So we use this clinically approved pump. This is the gene therapy in there. It doesn't seem very imposing, but it was hard to get that far. And then we just have tubes, and the tubes go into the catheter, and the catheter goes in the brain. And this is where the gene therapy goes. And as Dr. Bankiewicz said, he's been modifying this cannula so the solution will stay around where the tip of the infusion area, the tip of the needle, and it doesn't come up the needle. So by carefully modifying where this expansion point is, he can control where the solution goes. So this isn't this isn't Dr. Kildare. This isn't very exciting, but this is the practicality of it. We start with a very slow rate, and then every 10 minutes I increase the rate on the pump by 1 microliter per minute till I get up to 5 microliters a minute, and then we keep it at that rate. This is in the other gene therapy trial, because you have to start slow. If you start real fast, you can have some what's called retro flow or backflow of the fluid. So you have to start slow, let the brain tolerate the slower dose, and then you can gradually increase the rate. And this is our last patient that we treated, and here's the putamen, a much larger structure. So you can see it would be more difficult to cover completely compared to the much smaller substantia nigra and ventral tegmental area. But you could see that we covered, for the putamen, the total volume of this many cubic millimeters, we covered almost half of it here, which was pretty good with this, but because of substantia nigra and ventral tegmental areas are smaller, we should be able to cover those structures completely. And I just want to show a movie. This is what happens if we get MRI scans every 10 minutes. You get gradual expansion as you infuse for longer and longer, and you cover the area. And this was, Dr. Bankiewicz shared this with me. This was from the AADC trial at UCSF. So being in a clinical trial and being a patient is vastly different. Somebody comes in, they have back pain, I see them, I try to take care of them, but with a trial, you get included if you meet all the criteria. And it's not that we dislike some people, but it's just, that's just the way you have to standardize things, because the gene therapy will work better for a certain subset of people, and it will be safer for a certain group of people. So you want to give it to the people who might benefit the most. So So, but we're saying age 2 years to 16, but in the first 3 patients, there'll be at least 4. And I'm going to let Terry go through all that. I just wanted to make the point that I'm not a bad person, and if you're not included, it's not because of any personal animus. It's just that's the way trials are. So, and this is just an example. Here's for Parkinson's disease, this GDNF, glial-derived neurotrophic factor. 96 people inquired, and at the end of our first trial, our first dose, we only have 6 people that we've treated. So, and some of the people in this trial, we withdrew anyone who had any antibodies to Adeno-associated virus. So that was one of the reasons that some of those people were excluded. There are other reasons too. So it's in a trial, a lot of people in gene therapy, a lot of people don't want to have the lowest dose. So there's some reluctance. So here's the first person signed up in May of 2013. Then it took 9 months for the next person to sign up. But then you know, people talk, and then it's about every 2 or 3 months after that. And there will be more enthusiasm about getting higher doses. It's just this mindset that more is better. But maybe for effectiveness, maybe that's true. But for safety, you're probably better off at a lower dose. And the FDA believes that, our Food and Drug Administration. So we have to start at a lower dose. Now, so far with this gene therapy trial, these are our side effects. The first evening after the procedure, a lot of these people, they're old, they have Parkinson's disease. They had side effects from the anesthesia, and it might have been from the procedure too, but a few people had hallucinations. Some of the people were confused, but the most common complaints were headache and backache. And then during the first week after the procedure, headache, backache. They have backache because you have to lie on the table a long time. It takes a long time to aim the catheter. It's almost a whole working day procedure. And sometimes you get some bruising. We have to hold the head with a head holder, and they're sharp pins. So sometimes you get some bruising around that. That goes away. Long term, we really didn't see any specific side effects from the vector. And patients had symptoms typical of Parkinson's disease. Some of the people fell, but that's just part of the disease. You know, they'll have things like that. As far as efficacy at the lowest dose, some patients improved, some patients have not. And quality of life, some patients have improved, some have not. So our last patient was treated at the end of September, and we have this data safety and monitoring board. It will meet 3 months after that patient was treated and decide if it's safe to go to the next level. In this trial, we're going to put the viral vector in the substantia nigra, not in the putamen, as I mentioned before. And I'm going to let Dr. Pearson go through all this, but if you're in a, you know, everybody comes for the gene therapy, for the treatment, but in a clinical trial, there's a lot of other things that are included, you know, spinal taps, PET scans, lots of follow-ups, lots of trips to the hospital. So you have to be willing to do that too if you participate in gene therapy. Well, Dr. Bankwitz has taken the protocol through the preliminary steps, and he mentioned these: Office of Biotechnology Activities, Recombinant DNA Activities Committee, even the FDA, and got a investigational new drug approved. But this is one step we have to do. We have to get IRB approval at all the clinical steps. And you know, you might say, well, this is horrible. Why do we have all this regulation? It's just, they're just slowing things down. It's just a bunch of pencil pushers. But really, they want to only, all these boards want to approve only sensible, valid projects. And because the project's made it this far, it has that seal of approval, at least from those committees. The IRB will look at the ethical conduct of research and make sure that the informed consent document explains exactly what to expect and what the risks are. They want to protect human subjects, and you have to protect everybody because it is gene therapy. You don't want somebody to get the gene that's just that's a healthcare worker or the public. So you have to protect people other than the patients. And then once the trial's ongoing, there has to be protections for the patients, and the Data Safety and Monitoring Board looks at that, and the IRB reviews the protocol every year. So the studies to start sometime this year. Depends on a lot of factors, including how fast the IRB approves it and how fast we make the final revisions in the protocol. So thank you for your attention, and questions will be after the last speaker, so thank you.